Hey guys, welcome back to One Stop Biology. So guys, in today's video, we are going to cover the very second topic of chapter 12 of class 12th, which is biotechnology and its application. So in the last video, we actually went through, biotechnological applications in agriculture, and we saw how it has, greatly impacted the amount of produce and the quality and, a lot of things like say it controlled the pest that attacks the crops. It made the crops pest resistant crops, right? So in today's video, we are going to discuss the biotechnological application in medicine. Now guys, the biotechnology, the recombinant DNA technology has made a great impact. It has made immense impact in the area of healthcare. And how basically it has been implied is, it has enabled mass production of safe and effective therapeutic drugs, right? So it has not just done, wonders in the field of agriculture, but also in the field of medicine, right? So basically, if I talk about some numbers, at present there are about 30 recombinant therapeutics which have been approved from human use, right? And this is world over. And in India itself, 12 of them are being marketed, right? So let's see some of the examples. Now, the first example is of insulin, right? So genetically engineered insulin, right? So basically, for people who are suffering from diabetes, what happens is that they are given doses of insulin, right, at regular time intervals, right? So what happens is that, it has been extracted. The insulin which is used for diabetes was earlier extracted from pancreas of what? Slaughtered cattle and pigs, right? So basically, this caused the insulin which are extracted from these cattles or pigs used to cause patients with allergies or reactions to foreign protein, right? So basically, if you see the structure of insulin, insulin has two short peptides. It has two short polypeptide, right? So there are two chains, chain A and chain B. Okay. Now these two chains are linked together, right? So these two chains are linked together by a disulfide bridge, right? Now, happens that in mammals, including humans, insulin is synthesized as a prohormone, right? So basically, prohormone needs to be processed before it becomes a fully mature and functional hormone. And it contains an extra stretch which is known as C-peptide. So this insulin chain contains an extra stretch, and this stretch is known as C-peptide. Now, this C-peptide is not present in the mature insulin, right? So the main challenge for the production of insulin using the recombinant DNA technique was getting insulin assembled into a mature form, right? So basically what happened in 1983, Eli Lilly, an American company, prepared two DNA sequences corresponding to both the chains A and B, right? And chains of human insulin, and they introduced them in plasmids of E. coli. So they used plasmids of E. coli and they introduced the two DNA sequences corresponding to A and B chains. Okay, now chains A and B were produced separately and then they were extracted from this E. coli and it was combined by creating a disulfide bond, and we created a human insulin, a fully mature human insulin. So if you see the diagram here as well, you'll see that basically this is the chain of proinsulin. This is the proinsulin chain. And you can see it's the sulfur bonds as well, the disulfide bonds as well, right? So basically, this is the A peptide, and this one is the B peptide, right? And basically, this is the insulin. Two chains joined together. And this that you see is the free C peptide, right? So basically, C peptide is used to form a complete insulin, a mature insulin, right? This is the mature insulin. This is the proinsulin, right? So basically, what we did is we started making insulin genetically engineered, right? So we used recombinant DNA technique. What we did is we created two DNA sequences which were corresponding to A and B chains, and we introduced these chains into E. coli plasmid, and we produced chain A and B separately. We extracted it from the E. coli, and then we combined it by creating disulfide bonds, and we formed a mature human insulin without, remember, without hampering anything, right? In the same way, guys, recombinant DNA technology is used in gene therapy. Now we'll see what is gene therapy exactly. So, guys, basically, say if a person is born with a disease, with a hereditary disease, right? There is a collective therapy that we do. Right. Now, gene therapy again is a type of therapy against such diseases. And gene therapy is basically a collection of methods. And this collection of method allows correction of a gene defect that has been diagnosed in a child or embryo. Okay. So say we have a lot of tests that we do on an embryo. So when a mother is pregnant, what we do is we do a lot of tests to detect various type of hereditary diseases. Okay. And say if in a fetus, if in an embryo, such kind of disease is found, such kind of genetic disorder or hereditary disease is found, we try and cure it through a collection of methods which is known as gene therapy, right? So basically, to give, so say if I have to just define gene therapy is a collection of what? Collection of methods. And these methods allow what? Correction. Correction of a gene defect. Any genetic defect that has been diagnosed in a child or embryo. Okay, now how we do it? So basically, genes are inserted into a person's cell or tissue to treat a disease, right? So basically, correction of a genetic defect involves delivery of a normal gene into the individual or into the embryo to take over the functions of and compensate for the non-functional gene, right? So basically, we introduce a normal gene, and this normal gene takes over the function of a non-functional gene because of which there is a defect, right? Now, guys, the first clinical gene therapy was given to a 4-year-old girl in 1990. It was given to a 4-year-old girl in 1990. And that girl has a deficiency basically, which was adenosine deaminase deficiency. So basically, this is an enzyme which is crucial for immune system to function. Adenosine deaminase, ADA. This is an enzyme which is crucial for immune system to function. Now, because of this, there is a disorder which is caused due to deletion of the gene for adenosine deaminase. Right? So basically, in some children, this ADA deficiency can be cured by bone marrow transplantation. In others, in basically in patients with the same disease, it can be also cured by enzyme replacement therapy. Now, in that, functional ADA is given to the patient by injection. Right. But the problem with both of these approaches, whether it is bone marrow transplantation or enzyme replacement therapy, this is not basically these therapies will not completely cure the disease. These are not completely curative, right? And that is where we started off with gene therapy. So what we do is we take out lymphocyte from blood of the patient, and these are grown in a culture outside the body. So basically, we culture blood of a patient outside the body in vitro. We take out lymphocyte and we culture it. Then what we do is a functional ADA copy of a DNA, right, is introduced in those lymphocytes that we are culturing. And this was subsequently introduced in the patient, right? Now, since these cells are not immortal, they have a life cycle. The patient requires periodic infusion of such genetically engineered lymphocytes, right? But if we isolate the gene from marrow cell, which produces ADA, is introduced into the cells at early embryonic stages, it can be a permanent cure as well. So, say if now this ADA disease is detected during the embryonic stage itself, what we can do is we can just isolate the gene from marrow cells because in during the embryo stage it will start producing the lymphocytes, right? And we will introduce ADA at that particular time as well, which will permanently cure the disease. Okay, so such kind of gene therapy has evolved from recombinant DNA technology, from biotechnology basically, and this is helping a lot of patients with various hereditary disorders, hereditary diseases to be cured permanently, which is not possible from any other method, right? So this is all about gene therapy. Now the third process is molecular diagnosis, right? So basically, guys, right now we said that for the gene therapy to be started, we'll have to diagnose that particular disease as early as possible. So that means that for treatment, for effective treatment of a disease, early diagnosis and understanding of this physiology is very important, right? So say now conventional, if we talk about conventional method of diagnosis like urine analysis, serum analysis, blood analysis, right? Through these methods, early detection is not possible, right? But there are certain techniques which use recombinant DNA technology, like we have ELISA, which is a biotechnological technique basically, right? And this is enzyme linked immunosorbent assay. In the same way, we have PCR. PCR is also done. Polymerase chain reaction, right? So these are some techniques which basically help in early diagnosis. Right? Now, say, guys, presence of a pathogen, the pathogen can be a bacteria, it can be a virus, is normally suspected only when the pathogen has produced a symptom. So basically, we only get to know that we have COVID once our symptoms start. Once we start getting, itching in our throat, once we start getting fever, once we start getting congestion in our lungs and breathlessness because of that, right? In the same way, for any pathogen to be present in our body, we only realize that we have a disease once we have the symptom related to that disease, right? So by this time, we realize what happens is that concentration of pathogen is very high in the body, right? However, very low concentration of bacteria or virus can be detected by amplification of their nucleic acid by PCR. So basically, polymerase chain reaction is one method through which we can detect the presence of bacteria or virus when the concentration is very, very low as well, right? And that is why we use this process to detect, say, HIV in suspected AIDS patient. So even if they don't have, symptoms, but if we have, even a slight doubt that the patient might have HIV, might be HIV positive, we use ELISA, which uses PCR, right? Again, PCR is also being used to detect mutations in genes. And this mutation happens in cancer patients as well, right? Then we basically use it to identify a lot of genetic disorders, right? Now, what happens there is that, a single stranded DNA or RNA, we tag this strand with radioactive molecule, and then it is allowed to hybridize to its complementary DNA, right? So we basically then hybridize it to complementary DNA in a clone of cells, and then we detect it using autoradiography. Now, since the clone having mutated gene will not appear on the photographic film, why? Because the probe will not have complementarity with the mutated gene. And since we have tagged it, it is radioactive, we can detect that particular strand, right? So basically, in the similar way, ELISA, this test is based on the principle of antigen and antibody interaction, right? So infection by pathogen can be detected by the presence of antigen. If we have antigen, we can know that there is an infection of that pathogen. We can also detect it by presence of antibodies which are synthesized against the pathogen. So if you have heard, even in case of COVID, the test that we do is RT-PCR, right, which is a type of polymerase chain reaction, and antigen or antibody test. So basically, we either check for antigens if they are present in the body, we get to know that we have infection, or if we have antibodies against it. If our body is producing antibodies against the pathogen and we have antibody present in our body, that means that we have at certain point of time contacted against the pathogen and we were suffering from that particular disease, right? All these methods, guys, all these methods are nothing but biotechnological advances in medicine, right? So again, these are very, very important diagnostic methods that help us, right? These methods help us detect a lot of severe diseases which can be cured if detected at a very early stage. So guys, with this we have finished the biotechnological advancement in medicine as well. I hope you understood all these topics, all these small topics clearly. If yes, please do like the video and share it with all your friends. And if you're new to the channel, please do subscribe to the channel and press the bell icon to get all the updates that I'll be posting on my channel. And yes, guys, if you have any doubt in regards to the video, please do let me know your doubts either in the comments of this video and I'll answer it, or you can also message your doubts on WhatsApp, and the WhatsApp number is given in the description of the video. Thanks for watching this video, guys. See you in the next one. Take care. Bye bye.